Good morning. Thank you so much for inviting me to this talk. And good morning, session chairs, Professor Rahimi, Professor Wang, Professor Zeng, and Professor Jiang. Thank you so much. So today I will discuss our work recently published in Heart. The title of my talk is Role of Circulating Proteins in Cardiometabolic Diseases. So we recently published this paper in Heart. The title is Associations Between Circulating Proteins and Cardiometabolic Diseases, a systematic review and meta-analysis of observational studies and Mendelian randomization studies. This work is an international collaboration between University of Oxford, where I did my PhD degree, and Peak University, where I currently have my position in. So Professor Zheng Mingchen is my PhD supervisor, and Professor Liming Li is the current PI of our whole study group. Although this is only a meta-analysis, not an original piece of work, our team is currently conducting a lot of research looking at proteomics and cardiometabolic diseases. So my talk will be divided into 4 sections: introduction, methods, results, and our future work. As we all know, cardiometabolic disease, CMD, mainly includes coronary heart disease, stroke, and diabetes, and is the leading cause of death and disability in the world. One of the key research themes that our team is working on is called systematic epidemiology, 系统流行病学. It includes 3 key components: external exposure, which includes lifestyle and environmental factors; internal exposure, which includes metabolomics, proteomics, which are multi-omics biomarkers; and also clinical phenotypes, including both single diseases and multimorbidity. So proteomics can inform the holistic and comprehensive understanding of molecular and cellular mechanisms underlying the pathogenesis of diseases. And because proteins, they are key molecular biomarkers, and they are good candidates for drug target development. The objective of the study is to assess the direction and magnitude of the associations between circulating proteins and CMD, so as to provide clues and references for research on potential biological mechanism and drug target of CMD. Figure 1 shows the overview of analytical approaches and key findings of the study. So we did literature search, study selection, data extraction, meta-analysis, quality assessment, and comparison of risk prediction models, and downstream analysis. So the work can be divided into 2 sections. The first part is the observational analysis, looking at circulating proteomics biomarkers and the relationships with incident cardiometabolic diseases. So we can assess whether key proteins could improve risk prediction over traditional predictors such as smoking, alcohol, obesity. In the second part, we did Mendelian randomization studies. MR is a popular method for us to assess the causal relevance of proteins in the etiology of CMD. So key proteins might be potential candidates for drug development. Figure 2 shows the evidence grading for Mendelian randomization results. So we have 4 criteria: whether the reports of genetic association between protein and outcome was significant, whether the analysis was based on cis-pQTL only, whether the results passed multiple comparison, and also for proteins with more than 2 reports, we did a meta-analysis. So we divided proteins into 4 tiers. Tier 1 proteins refer to the protein with strongest evidence of causal association with diseases, and Tier 4 shows the weakest evidence. For meta-analysis, we conducted them separately for observational and Mendelian randomization studies, and we also reported separately each protein and disease pairs. So for the genetic analysis, we restricted to the European populations because that different populations have different genetic backgrounds, and trans-pQTL were excluded. So we're looking for consistent proteins, and a protein and disease pair was considered consistent if 2 criteria were met together. The first one is the association was significant in observational and genetic study and should be graded as tier 1 or tier 2 protein. And the second one is the directions of effect estimates were concordant in both the observational and Mendelian randomization studies. And this is the results. So in the observational study, we included 60 studies and identified 560 CMD-related proteins. In the Mendelian randomization studies, we involved 35 studies, and 245 proteins showed genetic associations, and they were either tier 1 or tier 2, showing the strongest evidence. In comparison, we observed only 22 pairs that showed consistent associations both in the observational and genetic analysis. And figure 4 is the so-called volcano plot for proteomics and diseases. And this is a comprehensive depiction of our entire association study. Next, we look at whether certain proteins would have pleiotropic effect, meaning one protein could have multiple functions. So in the observational studies, we saw 133 proteins showing associations with 2 or more CMD subtypes. For example, we observed 9 proteins showing associations with 4 diseases. In particular, 3 proteins showed associations with 5 diseases, which were GDF15, HAVR1, and MMP7. And in Mendelian randomization studies, we also identified pleiotropic proteins. 23 tier 1 or tier 2 proteins were associated with 2 or more CMD. Of note, 3 proteins showed genetic associations with 4 diseases. They were TMPS5, TNF12, TNR5. 1 protein, LPA, showed associations with 6 diseases, and 2 proteins, IL-6 receptor alpha and MMP12, showed associations with 7 diseases. Next, we looked at whether key proteins identified in our observational analysis, whether these key proteins would improve model performance over traditional cardiovascular risk factors. And in total, we identified 29 studies, including 79 models. So the number of proteins included in each model ranged from 1 to 291, and the median was 6 proteins. For model performance, we observed 13 models that could improve the C-index by 0.1 or greater. This is considered the clinical significance level of improvement. And also, 32 models showed a significant improvement over traditional model. And 28 models reached a C-index of 0.8 or greater. Also, we found 2 models, both for heart failure. The delta C-index over traditional model could reach as high as 0.19, and the 2 models both included 4 proteins listed here. Lastly, we evaluated the druggability of the key proteins, and we restricted to the 245 tier 1 or tier 2 proteins. We observed 3 protein pairs that had approved treatments, and the 3 proteins are both approved for treatment for heart failure because those 3 proteins are the drug targets to treat heart failure. Also, 288 protein, they have protein disease pairs that had no reports of drug development. This suggests that this protein might be candidate targets for future drug development. And also, 66 proteins had licensed drugs for indications different from the diseases implicated by our current study. This suggests opportunity for drug repurposing. And the table below showed the results from enrichment analysis. So we looked at different CMD subtypes and the significant pathways, both for GOBP and KEGG. And the figure on the left shows the detailed results. And you can see several key pathways were related to different subtypes of CMD, mainly for heart failure. For our future work, These are two papers published by our colleagues in Oxford. The first paper is looking at circulating protein biomarkers and ischemic heart disease and whether these key protein biomarkers could be the candidate targets to develop new drug. And the paper in the below section is an interesting paper looking at whether key proteins could be the drug target to tackle obesity. And lastly, this is our ongoing work looking at a wide range of proteomic biomarkers in relation to non-alcoholic fatty liver disease, NAFLD, showing the genetic association. And this is a comparison for adults in Chinese, East Asian, and also European population. And this is all for my talk. And thank you very much for your attention. And we look forward to submit more papers to HEART in the future. Thank you.